We've begun talking about exactly how it is. What is the actual physiology of the way the kidney does all of these amazing jobs that it does? And the place where it starts is here, at the renal corpuscle. The renal corpuscle has so much going on. It has got this area, the afferent arteriole, and wrapped around the afferent arteriole are these juxtaglomerular cells. Those juxtaglomerular cells do a lot. They are smooth muscle cells that will constrict or dilate the afferent arteriole in order to make sure that the pressure here inside the glomerulus is right in the sweet spot, no matter what your blood pressure is. Those juxtaglomerular cells are sensing blood pressure, and by being able to sense blood pressure, it allows them, when blood pressure is low, to send out that hormone renin. We have got the glomerulus itself and the glomerular capsule, both layers, that are going to be that wonderful filtration membrane. The efferent arteriole, we also have these things called mesangial cells and the macula densa that we aren't going to be talking about too much. Let's start talking about the glomerular capsule, though. The glomerular capsule's got two entirely different layers. This outer layer, known as the parietal layer, the outer layers of all of these structures gets called parietal. Right? The outer layer is just going to catch the fluid that is springing out of the holes here in the glomerulus and send that fluid down the very first part of the nephron tubule system called the proximal convoluted tubule. But this visceral layer, the visceral layer is composed of podocytes, and those podocytes create an extra layer of epithelial tissue, and they also have got an interdigitation of the pedicels on the podocytes, and those interdigitations create little slits in them that are called filtration slits. Inside of that, there's a layer of basement membrane, and inside of that, there are the simple squamous epithelial cells that make the structure of the capillary itself. And those simple squamous epithelial cells that, from this point of view, are the third layer down, those are not normal simple squamous epithelial cells. They are fenestrated themselves. So we are describing a filtration membrane, and a really sophisticated one. This is listing them from inside to out. So inside of here, there is blood going by, and the blood is looking at that layer that's depicted as pink here. And here we see a close-up of that pink layer. And the close-up of that pink layer, that is most of a simple squamous epithelial cell. And the simple squamous epithelial cells got little tiny holes in them that are called fenestrae, and that is a fenestrated epithelium. Now, those are little holes, and those holes are so small that blood cells, even platelets, are not small enough to sneak out through those holes. And that is why, even though there's lots of blood going through this structure that we're looking at here, the glomerulus, you will not normally find blood cells or even platelets out here in this filtrate. Okay. Now, the second layer. The second layer is basement membrane. That's no surprise. You've learned long ago in 150 that all epithelial cells, they've got their feet attached to basement membrane. Here's that basement membrane. And the basement membrane, it's going to be this mixture of proteins, collagen fibers, also proteins called proteoglycans, this kind of gooey matrix. And this messy structure excludes molecules that are even smaller. You don't have to memorize these numbers. I haven't. But now even some good-sized proteins cannot get through and get out into the filtrate. So you can see here, blood plasma is about 7% for protein, but this filtrate, much less than 0.1% protein. Why? Because the protein did not make it through the basement membrane layer. But that's not all. There's a third layer. And the third layer are these filtration slits, so these little interdigitations of the podocytes arms, the pedicels. Those create filtration slits, and they're negatively charged, And now even really tiny molecules can't get through. Here's what can get through. Write this down. Amino acids will end up out here, and glucose will end up out here. What else ends up out there in the filtrate? Water, tons of it. Urea, lots of it. Hydrogen ions, plenty of it. Bicarbonate, sure, that's out there too. Okay, lots of vitamins. Vitamins are out here. Water-soluble vitamins are out here. Lots of things end up out here in the filtrate. We're not going to pee those away. Okay, the glucose, the amino acids, the vitamins, we're not planning to pee those out. They are supposed to get reabsorbed before they end up in the urinary bladder. Okay. Let's talk about glomerular filtration rate. Glomerular filtration rate, or GFR, is one of the things that we will constantly be measuring in our kidney failure patients. Glomerular filtration rate, technically, is the total amount of fluid entering the capsular spaces of all the nephrons in each minute. So if I were to catch the fluid that is going out here into this Bowman's capsule and catch it for all of the Bowman's capsules in both of a patient's kidneys and catch all of that for 1 minute, that is the glomerular filtration rate. Glomerular filtration rate is kept pretty darn steady no matter what. If your blood pressure goes down, your afferent arteriole will dilate to keep the glomerular filtration rate steady. If your blood pressure goes up, the afferent arteriole will constrict to keep the glomerular filtration rate steady. Your kidney would like there to be a certain amount of this fluid to start out with. If you end up with too much glomerular filtration rate, then you'll end up making too much urine, and that will cause you to get dehydrated. If you start out with not enough glomerular filtrate per minute, then you will not be able to efficiently clean the blood. So this system is designed so that the glomerular filtration rate can be kept relatively steady, and a lot of that is by the relative diameters of the afferent and efferent arterioles. There is a typical glomerular filtration rate of about 125 milliliters per minute. That's about typical. And that is, if you calculate it all out, 1/2 of a cup every minute that you're alive. So 1/2 of a cup per minute. If I take 1/2 of a cup per minute, that's 1 cup for every 2 minutes. One of these videos is about 14 minutes, so that would be about 7 cups. That would be 1/2 gallon during one of these videos. 1/2 gallon. Can you imagine if you had to drink 1/2 gallon of liquid while you were doing a video? Well, of course you don't have to, right? Because you don't make 1/2 gallon of urine during one of these videos. Why? Because even though you start out during one of these videos, make 1/2 gallon of filtrate, about 99% of that is going to get reabsorbed. It goes back into your body where it belongs. So the system kind of seems really inefficient, right? We put all of this stuff out there and reabsorb 99% instead of just looking in the bloodstream and just grabbing the 1% and throwing it into the filtrate. Actually, birds and reptiles do that other thing, but we do this. Okay? This is something that I want you to know for your exam. What would happen if the glomerular filtration rate is not normal? What happens with GFR abnormalities? Inappropriate increases in GFR, and the biggest cause of this is high blood pressure. That will cause too much urine to be made. Why? Now, it's true that you can increase the amount of glomerular filtrate and end up with a normal amount of urine. You can do that. But there is a limit to how much can be reabsorbed from those tubules per minute. So if you're starting out with so much fluid going through that you simply can't reabsorb enough, you will end up with too much urine. And with too much urine, you can end up being dehydrated and have a depletion of what are known as electrolytes. I don't know if electrolyte is a chemistry term, but it certainly is a medical one. And when we talk about electrolytes, we are mostly talking about the small ions that are so critical for normal human physiology. The ions of sodium, potassium, calcium, chloride, phosphate, and bicarbonate. We're mostly talking about those. If you make too much urine, you can end up getting rid of too much sodium and end up with a sodium depletion, right? So that would be happening if you had too much GFR because like high blood pressure. But what happens if you don't have enough glomerular filtration rate? This is far more common. And when you don't have enough GFR, then wastes are not efficiently excreted, and you get a buildup of nitrogenous waste and a buildup of other waste products, right? So a decrease in GFR would happen if more than 2/3 of your nephrons have been destroyed, okay? So if you've lost half of your nephrons, then the rest of the nephrons, they increase their GFR so that the total GFR stays the same. But if you lose more than 2/3 or 3/4, then the ones that are left behind can't keep up, and you end up with a buildup of urea and creatinine and other bad stuff in the bloodstream. So how does the body regulate GFR? We will start that at the beginning of the next video.